Assalamu alaikum everyone. In this video lecture, we will be discussing the yearly past paper of AS level biology 9700. We will be discussing paper 2 and this is of October-November 21 paper 2-2. Let's start. Question number 1. There are two types of cell, prokaryotic and eukaryotic. Bacterial cells are prokaryotic and plant cells are eukaryotic. There are differences in the structural features that are common to bacterial cells and plant cells. For example, the cell surface membrane in plant cell contains cholesterol, but in a bacterial cell, the membrane contains molecules known as hopanoids. Cholesterol and hopanoids have the same function. Some of the main structural features common to both types of cells are shown in table 1.1. Complete table 1.1 by giving one difference between a bacterial cell and a plant cell for each structural feature listed. The difference between the cell surface membranes of the two types of cell has been completed for you. So guys, what we have to do, we have to differentiate the bacterial cell and the plant cell on the basis of the features listed. So cell surface membrane has already been done for us. We will look for the differences in the ribosomes. So if we talk about the ribosomes in the bacterial cell, so bacterial cell contains 70S ribosomes, right? Whereas if we talk about the plant cell, so plant cell contains 80S ribosomes. Plant cells also contain 70S ribosomes. You can also write that plant cells contain both 70S and 80S ribosomes. You can write that. But it's better to write that plant cells contain 80S ribosomes and bacterial cells contain 70S ribosomes because bacterial cells do not contain 80S, right? If you talk about DNA, so you all know that bacterial DNA is circular, right? So circular DNA in bacteria. And if we talk about plant cells, so plant cells contain linear DNA. You can also mention that the circular DNA that the bacteria contain lies freely in the cytoplasm, right? It's not surrounded by the nuclear envelope. Whereas if you talk about the linear DNA is found inside the nucleus. It's surrounded by nuclear envelope. You can also mention that. If you talk about cell wall, so bacterial cell wall is made up of peptidoglycan. Bacterial cell wall is made up of peptidoglycan or murine, right? And if we talk about plant cell wall, so plant cell wall is made up of cellulose. Plant cell wall is made up of cellulose. Right. Let's move on to part B. One role of the cell surface membrane of bacterial cells and plant cells is the transport of substances into and out of the cells. Explain how membrane carrier proteins and membrane channel proteins are involved in the transport of substances into and out of the cell. So guys, we have to mention that how carrier proteins and channel proteins, which are two types of transport proteins, are involved in the transport of substances into and out of the cell. So guys, you should know that carrier proteins are involved in active transport and facilitated diffusion, right? Whereas channel proteins are only involved in facilitated diffusion. You should know that carrier proteins have specific binding sites for specific molecules and carrier proteins change shape. So if they are carrying out facilitated diffusion, the shape changes passive just by the binding of molecules. Whereas if carrier proteins are carrying out active transport, so shape change requires the binding and breakdown of ATP, right? Channel proteins consist of channels or pores which are filled with water, or we can say these are hydrophilic pores which provide hydrophilic environment for the polar molecules and ions to pass. So guys, you should know that carrier proteins and channel proteins, they both are involved in the passage of polar molecules and ions. So we can write a lot of points over here. Let's write them down. We can first of all mention that channel proteins are hydrophilic, or we can say channel proteins contain pores filled with water. We can make an addition into this that channel proteins contain hydrophilic pores. hydrophilic pores filled with water that facilitate the passage of specific polar molecules and ions, right? We can mention that carrier proteins have binding sites for a specific polar molecule, for specific polar molecules or ions, and change shape or conformation to transport substances, right? You can also mention that channel proteins are involved in facilitated diffusion only, whereas carrier proteins are involved in both facilitated diffusion and active transport. Right. So we can also write some other points, but I guess for 3 marks, these points are enough. Let's move on to the next part, that is part C. Figure 1.1 is a photomicrograph showing chloroplasts in plant leaf cells. Alright, so guys, these are the plant leaf cells and we can see the chloroplasts over here. So these small structures are the chloroplasts, right? Alright. Explain why the chloroplasts are seen only around the periphery or edge of each plant cell. So guys, we can see that chloroplasts are near the edge of the plant cell. So why is that so? That's because there's a large central vacuole which is kind of turgid, right? Because this is filled with water. So there's a large central vacuole which is turgid, which is filled with water, and that pushes the chloroplast to the edges. So that's what we will be writing over here, that there's a large central vacuole. There is a large central vacuole which is filled with water and is turgid, so pushes the chloroplasts to the edge or periphery, right? Let's move on to part D. Figure 1.2 shows plant cells in a root tip where cell division by mitosis is taking place. So guys, this is the section of the root tip where mitosis of the plant cells is going on. Identify 2 cells in figure 1.2 that are in different stages of mitosis. Draw a label line to each cell and add the name of the stage of mitosis that is shown by the cell. So guys, what we have to do, we have to draw a label line to each cell and add the name of the stage of mitosis that is shown, right? So we just have to identify 2 cells in figure 1.2 that are in different stages of mitosis. So once we identify 2 cells which are in different stages of mitosis, what we have to do, we have to draw a label line and then we have to add the name of the stage of mitosis shown. So guys, if we look at this cell, we can see that chromosomes are in line. So this is definitely metaphase, right? If we look at this cell, we can see that chromosomes are scattered, and scattered chromosomes is the feature of prophase. So guys, this cell is in the prophase. If we talk about this cell is also in prophase. The chromosomes are scattered, right? So what we can do is that we just have to label two cells. So let's label this one and this one, and we will mention the phases. So this is prophase because chromosomes are scattered, and this is metaphase because chromosomes are in line. You all know that in metaphase chromosomes line up at the equator of the spindle, right? Okay, so we are done with question 1. Now we will move on to question number 2. The high blood pressure at the arterial end of capillary network results in some components of the blood leaving the capillaries and forming the tissue fluid. At the venous end, the presence of plasma proteins allow the movement of water by osmosis back into the capillaries. So guys, you all know about this information, that what happens is that this is the arterial end. Arterial end is the end of the capillary which is connected to arteriole. So basically this is the capillary, right? And this is also another capillary. This is the arteriole, and this end of the capillary, these ends of the capillary are known as the arterial end. So what happens when the blood enters the capillary at the arterial end? It is at high pressure, and what happens is that the fluid leaks out of the blood due to this high pressure, and the fluid that leaks out and fills the spaces between the cells of a tissue is known as a tissue fluid. So guys, the fluid that leaks out is the tissue fluid that I'm highlighting with green, right? So at arterial end, what is happening? The fluid is leaking out of the blood, and this is the tissue fluid. What happens at the venous end? Now, what is the venous end? Venous end is the end of the capillary near the venule. So this is the venule, right? And this is basically the end of the capillary, which is closer to the venule. So these ends are venous ends. What happens at the venous end is that the fluid, the tissue fluid, is returning back into the capillary. So why is that so? You should know that at the arterial end, what happens at the arterial end? Fluid leaks out of the blood and forms tissue fluid, right? So this is because of the high blood pressure. At the venous end, this fluid returns back to the capillary, and that is because there is high plasma protein concentration in the capillary, right? So in the capillary, in the blood, there is high plasma protein concentration. And why is that so? Why is there a high plasma protein concentration in the blood near the venous end or near the venule? That's because proteins do not leak out. When the fluid is being leaked out of the blood and this fluid is forming the tissue fluid, the proteins do not leak out, the large plasma proteins specifically. So what happens is that the fluid is leaking out, the water is leaking out, the small molecules are leaking out, leaving behind plasma proteins. So as the blood reaches the venous end, right? As the blood is reaching the venous end, the plasma protein concentration rises because the plasma or blood loses water but does not lose protein. So this causes the fluid to return back to the capillary by osmosis because the blood has a lower water potential at the venous end because of high plasma protein concentration, right? Do you get this point? At the venous end, in the blood, there is high plasma protein concentration, which causes the water potential to fall. As a result, the fluid moves back into the blood from a higher water potential in the tissue fluid to the lower water potential in the blood. And you should know that at the arterial end, what happens is that at the arterial end of the capillary, blood pressure is high, also water potential is also high, right? So let's move on to the questions. Red blood cells and plasma proteins such as albumin remain in the capillaries and are not found in tissue fluid. Explain why the red blood cells and albumin do not leave the capillary. So guys, as I mentioned earlier, that plasma proteins do not pass out or do not leak out of the capillary, and same goes for the red blood cells. That's because the red blood cells and plasma proteins are too large, right? Red blood cells and plasma proteins are too large to pass through the gaps or pores in the capillary endothelium. You know that if we talk about the capillary, so the wall of the capillary is made up of endothelium, which is a single layer of flat cells called endothelial cells, and there are gaps in the capillary endothelium. So guys, these gaps are very tiny, and what happens is that these gaps are actually very tiny, and red blood cells and albumin are too large to pass through these gaps, right? So these gaps are very tiny actually, right? So only small molecules can pass, and not only small molecules can cross, and not the red blood cells and albumin. So we will simply mention over here that the red blood cells and albumin molecules and albumin are too large. Okay, so do not pass through the gaps or pores in the capillary endothelium. Okay, let's move on to part B. Name the chemical reagent or reagents used to test for proteins in a sample of blood plasma, and state the color change that will be seen if protein is present. So guys, you all know that the reagent that is used to test for proteins is the biuret reagent. So the name of the reagent is the biuret reagent. Guys, please be careful, don't write it buret, it's biuret reagent. And the color change that we will see is the color change from blue to purple. B for biuret, B for blue. Biuret reagent is blue, and when proteins are present, the color changes from blue to purple. P for purple, P for proteins. So color changes from blue to purple. Right, let's move on to the next part. Part C. Okay, when a person moves from sea level and stays at high altitude. Guys, by the way, this question is no more in the syllabus. The concept of high altitude and the changes in the red blood cell number and the hemoglobin concentration is not the part of the syllabus anymore. So we will move on to part D straight away. A low blood albumin concentration can lead to a condition known as edema. Edema is a swelling of tissues caused by the accumulation of tissue fluid surrounding the body cells in the capillary network. Suggest and explain how a low blood albumin concentration can lead to edema. So guys, in this question they are giving us the information regarding edema. And what is edema? They are defining edema as the swelling of tissues caused by the accumulation of tissue fluid surrounding the body cells in the capillary network. So basically they are saying that normally what happens is that we just saw that tissue fluid is being evacuated. What happens is that the tissue fluid that leaks out at the arterial end returns back to the blood at the venous end or near the venule, and some of the tissue fluid also enters the lymph vessel and is evacuated. So almost all the tissue fluid is evacuated. You should know that. Edema is when there is excess tissue fluid in the tissues, maybe because there is some problem with the evacuation of the tissue fluid or excess tissue fluid is being formed. That is edema, right? So edema causes the swelling of tissues because there is excess tissue fluid in the tissues. And they are saying that low blood albumin concentration is one of the cause of edema. So we have to suggest and explain how low blood albumin concentration can lead to edema. So guys, let's come back to this point. I will erase this and I will restart it again because I have to explain that how low albumin concentration will cause edema. So guys, as we just saw that this is the arterial end of the capillary, because over here we have the arteriole. So what happens is that the fluid is leaking out at the arterial end from the blood in the blood capillaries, and the fluid which leaks out forms the tissue fluid. And if you remember what happens when the blood reaches the venous end. This is the venous end of the capillary, right? So what happens at the venous end or the end of the capillary near venule? The fluid returns back into the blood. The tissue fluid returns back into the blood. So as I mentioned, that what causes the fluid to return back into the blood at the venous end? This was low water potential caused by high plasma protein, right? So if somebody has albumin deficiency, albumin is one of the plasma proteins, right? So if someone has albumin deficiency, what will happen? There will be less protein in the blood, right? And the water potential will fall, but not significantly. So what happens is that the fluid returns to the blood at the venous end, but less fluid returns because the water potential falls in the blood, but not much significantly. There's a less steeper gradient between the tissue fluid and the blood at the venous end, right? Initially, there was very high protein concentration at the venous end in the blood, right? And because of this, water potential was very low. So a lot of fluid was dragged into the blood. A lot of tissue fluid was dragged into the blood. But now what happens is that protein concentration falls. So now there is less protein, right? And the water potential falls, but still the water potential does not fall much. So the gradient of fluid or water between the tissue fluid and blood is low. So less fluid is dragged into the blood. As a result, more fluid accumulates within the tissue, causing edema. Also, because there is low protein at the arterial end, so what will happen? This will cause the water potential to rise even more, right? We mentioned that at the arterial end there is high water potential and high blood pressure. So low protein at the arterial end will cause the water potential to rise even more. So more fluid will leak out at the arterial end. So what do we observe is that more tissue fluid is being formed, more fluid is leaked at the arterial end, and less fluid is evacuated at the venous end. So there will be accumulation of excess tissue fluid in the tissues causing edema. That's what we have to write. So what will we write over here? Low albumin concentration in the blood means less protein concentration in the blood, right? Low albumin concentration in the blood means less protein concentration in the blood, right? So there is a steeper concentration gradient, or steeper, we can say steeper water potential gradient. Water potential gradient at the arterial end, right? So there is a steeper water potential gradient at the arterial end, so more tissue fluid leaks out. So there is a steeper water potential gradient at the arterial end, so more tissue fluid leaks out or is forced out. You will also mention that there is a less steep water potential gradient at the venous end of the blood capillary, so less fluid moves into, so less tissue fluid moves into the blood, by osmosis, right? We can also mention that low albumin concentration in the blood means less protein in the blood and higher water potential of blood. Okay. Albumin transports some cell signaling molecules from the cells where they are synthesized to their target cells. The cell signaling molecules bind to specific cell surface membrane proteins on the target cells. Name the type of membrane protein to which the signaling molecules bind. So guys, they're mentioning over here that albumin is a plasma protein that transports signaling molecules. Actually, albumin binds these signaling molecules, and albumin transports these signaling molecules in the blood from where they are synthesized to their target cells. Basically, in this question, they're asking us the type of protein in the membrane to which the signaling molecule binds. And you all know that signaling molecule binds to a receptor. This receptor could be in the cell surface membrane or could be inside the cell. So guys, we are done with the question 2. Now we will move on to the next question.